What does it mean to build with life? How do we reconnect with the circular flows from which life evolved? Emerging from methane plumes in the ocean 4.5 billion years ago, the first known living cells formed the foundation of an increasingly complex web of life, coding and decoding DNA, living and dying, growing and decaying. The ocean food web is under threat from 80 years of plastic production made from fossilized life extracted from the earth. This material used for the disposable products never degrades, but breaks down into microplastics which circulates throughout the planet. The persistence of microplastics within our ocean is a prevailing issue researchers are trying to solve. We set out to design a floating research center to aid scientists in the remediation of this problem. Our design integrates polyhydroxyalkanoates, or PHA, a plastic that breaks down into fish food, as the main structural component of the architecture. These floating structures, called living pods, will be located near urban shorelines where multidisciplinary teams can utilize the information gathered from them. The San Francisco Bay Microplastics Report, published last fall, issued recommendations for policy actions. One was to work with local universities to prioritize research into microplastic pollution in the San Francisco Bay. Our project is a response to this call to action. The story begins with a simple gas, methane, an elemental hydrocarbon that occurs naturally in places like thermal vents, deep in the ocean, or under frozen lakes. This greenhouse gas is 25 times more potent than CO2. Yet a bacteria in the methanotrope family exploit this gas as a resource. This ancient bacteria uses methane to produce a fat known as polyhydroxyalkanoates, PHA. Our project synthesizes PHA biopolymers with large-scale emerging robotic 3D printing methods to fabricate floating multidisciplinary research vessels. Research is needed in the open ocean to study the relationship between the PHA thickness and the rate of degradation. In Phase 1, the small pod will be fabricated to support low-cost deployable sensors powered by wind. It will act as a buoy, revealing the damage being done by plastic to our marine environment. In Phase 2, we'll take lessons learned from the degradation of the small pod, make adjustments, and scale up to a medium-sized pod designed to work with the currents. It will be deployed where the floating plastic accumulates. The parametrically shaped fins pull surface debris into the center where it is collected and taken back to the main pod to be categorized and publicly viewed. In Phase 3, we'll take lessons learned and further refine the design to serve as a public classroom with a wind and wave-powered interior projection. This pod also showcases plastic remediation exhibits and experiments which connect ocean art and science. In 1947, a wooden craft called the Kon-Tiki, made of balsa logs, sailed from Peru to Polynesia. Wood log density is lighter than seawater. This keeps it buoyant and appropriate for marine application. In the Bay Area, logs are easily sourced and sequesters carbon. The platform forms a flotation device for the living pod. The logs are arranged to direct flows into the center to collect surface debris. In the first generation of the design, we printed 3D models with a corn-based bioplastic called PLA and tested it in the ocean. After many months, the colonization of the prototype was amazing. Surface differentials of hills and valleys successfully created a habitat for a variety of invertebrates, even tiny shrimp. But the material didn't break down. This insight that even the corrosive ocean would not break down PLA inspired us to reach out to Molly Morse, the founder of Mango Materials, a company that is at the forefront of bringing PHA to the marketplace. Molly explained that in the ocean, microorganisms will consume the PHA and completely break it down to the end carbon elements that are not only non-toxic but food. This became the driver of the design. How do we design a 3 to 5 year demonstration degradation project? How thick does the shell need to be to meet the project needs? How thin does it need to be to function as a tissue which breaks down to activate the pod to open and move with gravity towards the seabed? And as it drops to the ocean floor, how do the parts with marine life attached and feeding on its shell become a new ecological reef helping to slow coastal storm surges? We kicked off the 2020 Biodesign course with a visit to the Prescott School 5th grade classroom. California College of the Arts architectural students shared invertebrate drawings. Their teacher said, when a student learns something new, then draws or makes it, they now own it. It becomes their own. 3 scales of living pods work together to form an interconnected system to educate the public and study the catastrophic damage of plastic that has done to the ocean. The study revealed that microplastic levels in the bay's surface waters were higher than other urban embayments and were linked directly to single-use plastics. The design of the pods channeled the surface water into the center, creating a passive collection for these floating plastics, which can be regularly removed by student researchers and used to educate the public on humans' role in what is floating in the bay. The small living pods are interspersed around the bay. They use a novel low-cost Plankton Scope recently developed by Dr. Manu Prakash at Stanford University. The Plankton Scope samples seawater at programmable intervals to understand the diversity of microscopic organisms as well as quantify microplastic debris. They create a network of remote research devices that use artificial intelligence to sort and relay information to science labs, classrooms, cell phones, and real-time projections within the shell of the largest pod, located off the Embarcadero Shoreline Walkway, where millions of people circulate each year. On the deck, people can learn about speculative biodesign approaches to the systemic remediation of larger systems such as Elizabeth Marley's thesis work from the University of California, Davis, and see biodegradation experiments and a rotating taxonomy of collected single-use plastics from the mid-size pod.